All right, settle in, because today we're embarking on a deep dive into something truly fundamental. We're talking about the very blueprint of life in the plant kingdom. From, the subtle patterns on a corn kernel to the
Yeah.
unseen molecular machinery driving growth. We're going to unpack the incredible world of genetics and evolution in plants.
Indeed. We'll explore how traits are passed down, how those traits are expressed at a molecular level, and crucially, how these processes drive the incredible diversity and adaptation we see all around us. Our mission today, basically, is to cut through the complexity. We want to give you a clear, comprehensive understanding of these core concepts, drawing insights directly from Raven biology of plants.
Think of it as a journey. We start with a microscopic, intricate dance of chromosomes
Right.
and end up at the grand, sweeping narrative of evolutionary change. We've got a stack of fascinating material to get through, and we promise you some genuine aha moments. Might just change how you look at the nearest leafy green. Let's start right at the beginning then, with sexual reproduction. It's a defining characteristic for most eukaryotes, plants included, and it involves this really intricate ballet of two critical processes.
Meiosis and fertilization.
Exactly. It's how life, especially complex life, continues from one generation to the next.
That's right. Meiosis is this specialized nuclear division. Its job is to have the chromosome number. It takes a diploid cell, meaning it has two sets of chromosomes, usually written as 2N,
Okay, two set.
and produces 4 haploid nuclei, each with just one set, or N. In plants, this is how they make spores, maybe in flowers or, cones.
Right.
Then comes fertilization, sometimes called syngamy. It's basically the fusion of two of those haploid cells, the gametes, to form a new diploid zygote. And boom, you've restored that 2N chromosome number.
Ah, so it resets the count.
Exactly. This constant alternation ensures the chromosome count remains stable across generations. It's absolutely essential for species stability.
So it's like a genetic handshake. It ensures stability, but it also creates new combinations.
Precisely.
And in every diploid cell, those chromosomes aren't just floating around randomly. They have partners.
That's a really crucial distinction. These are homologous chromosomes, or homologs. They match up size, shape, and critically, they carry similar hereditary information. Think of them as pairs.
Okay.
One homolog comes from each parent's gamete. After fertilization, both are present in the zygote, and their genes interact. That interaction determines the organism's characteristics. This precise pairing is the foundation for, well, everything else we'll talk about regarding segregation and recombination. It sets the stage for genetic diversity.
Now let's zoom in on the chromosome itself, because it's not just a simple string of DNA, is it?
No, far from it.
We're talking about an incredible amount of information packed into a truly tiny space.
It's amazing, really. Eukaryotic chromosomes are incredibly complex structures. They're made of DNA, of course, but also associated proteins, mainly histones. This whole combination is called chromatin. The name actually comes from its property of staining readily. You can see these colored threads under a microscope.
Right.
Now, histones are small proteins, and they have a positive charge. DNA, on the other hand, is negatively charged.
So they attract each other.
Exactly. Opposites attract. And they're absolutely essential for DNA's meticulous folding and packaging. Imagine our DNA, this microscopic thread, being wound tightly around tiny protein spools.
Like thread on a spool.
Yeah. Each spool, called a nucleosome, consists of a core of 8 histone molecules with the DNA filament wrapped around it twice. Then another histone, H1, sort of helps lock this into place, like a clamp. It further compacts the structure.
Giving that bead on a string appearance you sometimes hear about.
That's the one. It's a great visual.
And how much DNA are we talking about here to need such elaborate packaging? What's the scale of this challenge?
Oh, it's a truly staggering amount.
Yeah.
For instance, a single diploid cell of the wake robin flower, Trillium erectum,
Okay.
contains about 68 meters of DNA.
68 meters.
Yep. That's a remarkable length to fit into a nucleus that's only a few micrometers across.
That's incredible packaging.
It really is, and that beads on a string structure is just the first level. It then folds further, coiling into a denser fiber, forming larger loops, and finally condenses into the compact metaphase chromosomes that we can actually see with a light microscope during cell division.
So it's multi-level packing.
Exactly. And it's vital not just for fitting the DNA in, but also for regulating which genes are accessible and when. It's a marvel of biological engineering, controlling both space and function.
This intricate packaging must be absolutely crucial for meiosis then. That's the process that really shakes up the genetic deck, right?
Yeah.
This is where the profound variation we see in offspring actually begins. So what's happening in those key meiotic stages to make that diversity possible?
Okay, so meiosis happens only in specialized diploid cells. And it's a two-stage process, very different from mitosis, which just makes identical copies.
Right, growth and repair versus reproduction.
Exactly. In prophase I, the first stage, chromosomes condense, become visible. And critically, homologous chromosomes pair up precisely. Imagine two zippers coming together, matching tooth for tooth. This forms a bivalent that's a homologous pair made of 4 chromatids in total.
Okay, 4 strands together.
Yes, and here's where a truly crucial event for genetic variation occurs. Crossing over. Portions of these chromatids physically break and rejoin with their homologous partners. They literally exchange genetic segments.
That's Rex swap.
A direct swap. This results in chromatids with entirely new gene combinations. You can even see visible evidence of this under a microscope, these X-like configurations called chiasmata. That's where the swap is physically happening.
Right.
And the further apart 2 genes are on a chromosome, the more likely it is that a crossover event will happen between them.
So a physical swap of genetic material between the paternal and maternal contributions. That's where the real mixing happens.
Absolutely. It ensures that, barring identical twins, no 2 offspring are truly genetically identical.
Okay, what happens next?
Then, in metaphase 1, these paired homologous chromosomes line up randomly at the cell's center. This random alignment itself is another major source of genetic variation.
Independent assortment.
That's it, independent assortment. How one pair lines up doesn't affect how another pair lines up. Then, in anaphase 1, the paired homologous chromosomes separate and move to opposite poles. But importantly, each chromosome still consists of 2 sister chromatids at this point.
Okay.
Meiosis II then proceeds much like mitosis. In metaphase 2, chromosomes align at the equator again. In anaphase 2, the centromeres finally separate and the sister chromatids move to opposite poles. Finally, telophase 2 results in 4 haploid nuclei, each genetically unique.
Unique because of crossing over in that random alignment.
Precisely. These nuclei will then develop into gametes or spores, depending on the plant.
And just to be clear, how does this elaborate 2-step process compare to mitosis? What are the key takeaways?
Right, good question. 3 fundamental differences really highlight meiosis's unique role. First, meiosis involves 2 nuclear divisions, but only 1 DNA replication. Mitosis is 1 division, 1 replication.
Okay, 2 divisions, 1 copy job.
Yep. Second, meiosis produces 4 haploid nuclei, each with half the chromosomes. Mitosis produces 2 diploid nuclei, identical to the original cell.
Half the chromosomes versus the full set.
Exactly. And third, and this is critical for evolution, each meiotic product has unique gene combinations due to that crossing over we talked about, and the independent assortment of homologous chromosomes.
Whereas mitosis makes clones.
Right. Mitotic products are genetically identical.
Yeah.
This genetic reshuffling in meiosis is profoundly important. It's the engine that creates diverse populations, increasing the chances that some individuals will be better equipped to survive changing environments.
Okay, so the physical basis of heredity is meiosis, shuffling those genes. But the foundational patterns of inheritance were famously cracked by Gregor Mendel, right? The 19th century monk with his pea plants.
Yes. His work was truly brilliant, especially considering nobody knew about DNA or chromosomes back then.
It's still astonishingly relevant.
Absolutely. Mendel chose pea plants, Pisum sativum, very carefully. He picked ones with clearly contrasting traits, yellow versus green seeds, purple versus white flowers, things like that. And crucially, he didn't just look at the first generation. He performed large numbers of crosses, meticulously tracking traits, not just in the first generation, the F1, but also in subsequent ones, F2.
Patience and good record keeping.
Immense patience. He observed that in the F1 generation, one trait would often disappear, only to reappear in the F2 generation in predictable ratios, like 3:1. He called the visible F1 trait dominant and the hidden one recessive. His success was also greatly helped by the fact that pea plants normally self-pollinate. This allowed him to control crosses precisely and ensure he started with true breeding lines, plants that always produce offspring like themselves.
And from all this careful work, he developed 2 foundational principles. Still cornerstones of genetics today, aren't they?
They absolutely are. First, the principle of segregation.
Okay.
This states that hereditary characteristics are determined by discrete factors, what we now call genes, that exist in pairs. One factor comes from each parent. During meiosis, when gametes are formed, these pairs separate or segregate. So each gamete receives only one member of the pair.
Ah, so that explains how a recessive trait like green seeds could vanish in F1 but pop up again in F2.
Precisely. It explains how traits persist without blending and why a trait might seemingly skip a generation.
And the second principle.
The principle of independent assortment. This states that the two alleles for one gene sort independently of the alleles for other genes during gamete formation. Meaning, the inheritance of seed color, for example, doesn't affect the inheritance of seed shape. They sort into gametes independently of each other, assuming they're on different chromosomes or far apart on the same one.
Okay, got it. He also gave us the Punnett square, didn't he? That diagram for predicting offspring?
Yes, the Punnett square, a simple visual tool to predict the genotypes and phenotypes of offspring from a cross, and the testcross.
The testcross.
Yeah, where you cross an individual showing a dominant trait, but whose genotype you don't know. It could be homozygous dominant or heterozygous.
Right.
You cross it with a homozygous recessive individual. The results tell you the unknown genotype. Still fundamental tools in genetics labs and classrooms. Now, while Mendel's principles are fundamental, genetics, like life, has its nuances. It gets more complex.
Okay.
Linkage, for example. This explains that if two genes are located physically close together on the same chromosome, they often don't assort independently.
So Mendel's second law isn't always true.
It holds for genes on different chromosomes or far apart, but not when they're close together. These nearby genes are linked. They tend to be inherited together as a unit because crossing over between them is less likely.
The closer they are, the tighter the link.
Exactly. This was first discovered in sweet peas, actually, by William Bateson and his colleagues back in 1905. They saw trait ratios that just didn't fit independent assortment. What's really cool is that the frequency of crossing over between linked genes, how often they do get separated, can be used to map their relative positions on the chromosome.
Like creating a genetic roadmap?
Precisely. These are called linkage maps.
Okay, so physical location matters. What about the ultimate source of all this genetic variation? Where do completely new traits come from?
Okay.
Mutations, right?
Mutations, yes. They are simply changes in an organism's hereditary makeup, the DNA sequence. They are the absolute raw material for evolution.
Where did the term come from?
Hugo de Vries coined it. He observed sudden, unexpected new traits appearing in evening primroses. Today, we recognize several types. You have point mutations, which are small changes at the level of a single gene, maybe substituting one DNA base for another.
Like a typo.
Sort of, yeah. Then you have larger scale chromosome mutations. These affect chromosome structure. You can have deletions, where segments are lost, duplications, where segments get doubled, inversions, where a segment flips 180 degrees, and translocations, where segments get swapped between non-homologous chromosomes. Big rearrangements.
Okay.
And then here's where it gets really interesting, in my opinion. Transposons.
Jumping genes.
Exactly. Barbara McClintock's Nobel Prize-winning work in maize, those beautiful multicolored corn kernels you see.
Yeah.
She showed that segments of DNA can literally move or jump around the genome. These transposons, sometimes called molecular parasites, can insert themselves into or near other genes. This can disrupt the gene or change how it's expressed, what are called position effects, leading to new characteristics.
So mutations, including these jumping genes, provide the brand new variations.
They're the ultimate source, yes. They provide the raw material that natural selection can then act upon, allowing populations to adapt over time. Without mutation, evolution would eventually grind to a halt.
Okay, so it sounds like the story of how genes actually work, how they create traits, is much richer and more intricate than Mendel first imagined. There's more going on than just simple dominant and recessive.
Oh, definitely. Many traits don't fit those simple patterns. For instance, we commonly see things like incomplete dominance.
What's that?
Remember the snapdragons? Cross a red one with a white one, and you don't get red or white offspring in the F1 generation, you get pink.
Ah, a blend.
Exactly. The heterozygote shows an intermediate phenotype, a blend of the two parental traits. Neither allele is fully dominant.
Okay, what else?
Multiple alleles. Even though any single diploid organism can only carry 2 alleles for a gene within a whole population, there can be many more than 2 forms of that gene. Self-sterility genes in red clover, for instance, can have hundreds of different alleles circulating in the population. It's amazing.
Wow, hundreds. Okay.
Then there's polygenic inheritance. Many traits we care about, like size, shape, or yield in crops, aren't controlled by just 1 or 2 genes. They're the result of many genes interacting, each contributing a small amount to the final outcome.
So it's additive.
Often, yes. And these traits typically show continuous variation, a smooth gradation of phenotypes. If you plot the distribution in a population, you often get a bell-shaped curve. Think about human height or ear length in maize. It's not just tall or short, but a whole range.
Right, unlike Mendel's distinct categories.
Exactly. And finally, gene interactions, specifically something called epistasis.
Pick.
This is where one gene can interfere with or mask the effect of another gene at a completely different location, a different locus. In foxglove, for example, there is a gene that restricts pigment to just spots on the throat. This gene is epistatic to other genes that determine whether the main petal color should be dark or light red.
So the spot gene overrides the color genes.
Pretty much, yeah. It masks their effect on most of the flower, so the genetic interactions themselves can be complex.
Okay, so the internal genetics are complicated.
Yeah.
But the environment plays a role too, right? It's not just about the DNA sequence.
Absolutely critical point. The phenotype, what we actually see, the observable characteristic, is always an interaction between the genotype and the environment. Always.
Nature and nurture interacting.
Precisely. A plant might have the genes to be green, but if you keep it in the dark, it won't produce chlorophyll, so it won't be green. A really striking example is the water buttercup, Ranunculus peltatus. It grows partly submerged, partly floating. The leaves are genetically identical, same DNA, but the underwater ones are finely divided, almost feathery, while the ones floating on the surface are broad and flat, like typical leaves.
Wow, same genes, totally different look.
Totally different. It's due to environmental cues affecting how the cells expand and develop, not differences in the genes themselves. It highlights this amazing plasticity plants have.
That's fascinating. Anything else influencing inheritance?
Yes. One more thing, we also see maternal inheritance for traits determined by genes in plastids, like chloroplasts and mitochondria.
Because those organelles often come just from the egg cell.
Exactly. Sperm cells usually don't contribute much cytoplasm or organelles during fertilization, so these characteristics are typically passed down solely from the female parent.
We've focused so much on sexual reproduction and all the genetic diversity it creates. But some plants take a completely different route. Asexual reproduction, what's going on there?
Right. It's also called vegetative reproduction. This produces progeny that are genetically identical to the single parent. They're clones.
No mixing of genes?
No mixing. Meiosis and fertilization are absent. It's all based on mitosis, just making identical cell copies. Plants have many ingenious ways to do this. Think about runners on a strawberry plant, or bulbs, or even just stems rooting if they touch the ground.
Yeah.
There's also something called apomixis. This is where embryos in seeds are produced asexually without fertilization. So you get seeds, but they contain clones of the mother plant. Dandelions do this, so does Kentucky bluegrass.
Seeds without sex?
Pretty much. And many species can actually do both sexual and asexual reproduction. They can hedge their evolutionary bets depending on the conditions.
That makes sense. If a plant has a winning combination of genes for its current spot, why wouldn't asexual reproduction just take over? What are the trade-offs?
That's the core evolutionary dilemma, isn't it? Asexual reproduction is incredibly efficient. You replicate individuals perfectly suited to a stable environment. If you've got a winning genetic hand, why shuffle the deck?
Right.
However, and this is the huge however, without the recombination and genetic variability generated by sex, these populations struggle, often badly, to adapt to changing conditions.
Like new diseases or climate change.
Exactly. A new disease could wipe out an entire clonal population if they all have the same susceptibility. Sexual reproduction, while energetically costly, you need flowers, pollinators, gametes.
It takes effort.
It takes effort, but it produces that rich genetic diversity. This diversity means it's much more likely that some offspring will have novel combinations of alleles that allow them to survive new challenges.
So sex is an investment in future adaptability.
That's a great way to put it. The sheer prevalence of sexual reproduction among eukaryotes today strongly suggests its long-term evolutionary advantage despite the short-term costs.
Okay, let's shift gears now. Moving from inheriting traits to the molecules themselves. Chapter 9 dives into the chemistry of heredity and the discovery of DNA structure. Well, that was a total game changer, wasn't it?
Oh, absolutely pivotal. A landmark moment in biology. In the early 1950s, scientists knew chromosomes carried genetic information, but there was this big debate. Was it the DNA or the protein in the chromosomes that held the code?
Proteins seem more complex, maybe.
Exactly. Proteins have 20 different amino acid building blocks. DNA only has 4 nucleotide bases. So proteins seem like a better candidate for complex information. But it was James Watson and Francis Crick crucially synthesizing data from many others. Rosalind Franklin, Maurice Wilkins, Erwin Chargaff.
Right, building on others' work.
Absolutely. They finally deduced the structure in 1953. They realized DNA wasn't a single helix, but an entwined double helix. Imagine a ladder, right? Then twist it into a spiral. The sides of the ladder are made of alternating sugar deoxyribose and phosphate groups,
Okay.
and the rungs of the ladder are formed by pairs of nitrogenous bases.
Adenine, thymine, guanine, cytosine, ATGC.
Those are the ones. And Rosalind Franklin's X-ray diffraction images, her famous Photo 51, were absolutely crucial in figuring out it was a helix and getting the dimensions right.
And those bases, the rungs, they had a very specific pairing rule, didn't they?
Yes, this was another key insight, building on Chargaff's earlier findings. Chargaff had noticed that in any DNA sample, the amount of adenine A always equaled the amount of thymine T, and the amount of guanine G always equaled cytosine C.
ATGC,
Yeah.
Chargaff's rules.
Right. Watson and Crick realized why. An A on one strand could only pair with a T on the opposite strand, forming 2 hydrogen bonds, and a G could only pair with a C, forming 3 hydrogen bonds.
Specific pairing.
Specific pairing. This ensures the 2 strands are complementary. If you know the sequence of bases on one strand, you automatically know the sequence on the other.
Like a template.
Exactly. And also, the 2 strands run in opposite directions. They're antiparallel. If one runs, say, top to bottom in a chemical sense, 5' to 3', the other runs bottom to top, 3' to 5'.
Okay.
This anti-parallel structure, combined with a specific base pairing, immediately suggested to them, in their famous understated paper, a possible copying mechanism for the genetic material.
Understatement of the century.
Uh.
Yeah, that complementarity just screams, copy me.
Mm-mm.
It immediately suggests how DNA makes copies of itself, which is absolutely essential for passing on hereditary information.
It does. During DNA replication, the double helix locally unzips. Enzymes break the hydrogen bonds, holding the base pairs together. Each separated strand then acts as a template, a blueprint for building a new complementary strand.
Using those base pairing rules.
Exactly. Free nucleotides floating in the cell nucleus pair up according to the strict ATGC rules. A matches T, G matches C, and specialized enzymes called DNA polymerases catalyze the reaction, adding these nucleotides one by one to the growing new strand.
Creating 2 new helices from one.
Right. This results in 2 exact replicas of the original DNA molecule. And importantly, each replica contains 1 strand from the original molecule and 1 newly synthesized strand.
Semi-conservative replication.
That's the term, semi-conservative. It was a beautiful solution to how hereditary information could be copied so accurately generation after generation.
Okay, so that's the principle. But how does this happen in a large, complex eukaryotic chromosome? You mentioned meters of DNA earlier. It can't just unzip from one end to the other, can it?
No, that would take far too long. It's a highly complex and coordinated effort, like a molecular pit crew.
Okay.
Replication doesn't start at the ends. It begins at multiple specific spots along the DNA called origins of replication. If you look with an electron microscope, you can actually see these regions forming replication bubbles. They look like eyes opening up along the DNA strand.
Multiple bubbles opening at once.
Yes, simultaneously at many origins. At each end of a bubble, there's a Y-shaped structure called a replication fork. That's where the parental strands are actively unwinding and the new strands are being synthesized.
And these forks move.
They move away from the origin. In opposite directions, it's bidirectional replication. The bubble expands until it merges with an adjacent bubble. Loads of enzymes are involved. Helicases unwind the DNA. Other proteins keep the single strands from repairing. Topoisomerases prevent tangling further down the helix.
A whole team.
A whole team. And there's another complication. DNA polymerase can only build a new strand in one specific chemical direction.
5 to 3.
Okay, so what does that mean at the fork?
It means one new strand, called the leading strand, can be synthesized continuously, smoothly, following the unwinding fork.
Easy enough.
But the other strand, the lagging strand, has to be synthesized discontinuously. It's made in short segments, backwards relative to the fork's movement. These are called Okazaki fragments.
Little pieces made backwards.
Yeah, kind of stitched together later by another enzyme, DNA ligase, which acts like a molecular glue. It's an incredibly elegant, if complex, solution to the enzyme's directional limitation. Okay, so we have the DNA, accurately copied. Now, how does that information, that sequence of A's, T's, G's, and C's actually get expressed? How does it become a trait, like flower color or plant height? How does it build the proteins that do all the work?
Right. This is the next crucial step, often called the central dogma of molecular biology. The flow of information is generally DNA is transcribed into RNA, and then RNA is translated into protein.
DNA to RNA to protein.
That's the core flow. RNA, ribonucleic acid, is DNA's close cousin, its sister molecule. It's similar, but with a few key differences. It uses ribose sugar instead of deoxyribose. It uses the base uracil U instead of thymine T, so U pairs with A. And it's usually single-stranded, not a double helix.
And it acts as the messenger.
It acts as the essential intermediary, carrying the instructions from the DNA blueprint out to the protein-building machinery.
So DNA stays safe in the nucleus, mostly, but RNA can move out.
Exactly. Most RNA is found out in the cytoplasm, which is where proteins are actually synthesized. There are three main types involved in protein synthesis. You have messenger RNA, mRNA, which carries the actual genetic code, the message, from a gene.
Okay, the messenger.
Then transfer RNA, tRNA. These act as adapters. They carry specific amino acids, the building blocks of proteins, to the ribosome.
The delivery trucks?
Kind of, yeah. And ribosomal RNA, rRNA, which is a major component, structural and catalytic, of the ribosomes themselves. Ribosomes are the protein factories.
Okay, mRNA, tRNA, rRNA. And the famous genetic code, how does the mRNA sequence specify which amino acid goes where?
The genetic code is read in triplets. Sequences of 3 consecutive nucleotides on the mRNA are called codons. Each codon specifies a particular amino acid.
3 letters make a word.
Exactly. With 4 bases, A, U, G, C, reading them 3 at a time gives you 4 by 4 by 4 by 64 possible codons. Since there are only 20 common amino acids used to build proteins, there's some redundancy in the code.
More than one codon for the same amino acid.
Yes, many amino acids are specified by more than one codon. Often the first 2 letters are the same and only the third one differs. This actually provides a bit of a buffer against some mutations. There are also specific start codons, usually AUG, and stop codons that signal where protein synthesis should begin and end.
And this code is universal.
Remarkably so. With only very minor exceptions, the same codons specify the same amino acids in virtually all organisms, from bacteria to archaea to plants to humans.
Wow.
It's incredibly powerful evidence for a common ancestor for all life on Earth. And it's the foundation that makes genetic engineering possible, putting a human gene into bacteria, for example.
Okay, so how did transcription and translation, these 2 big steps, actually unfold?
Right. First, transcription. This happens in the nucleus in eukaryotes. An enzyme called RNA polymerase binds near a gene on the DNA. It unwinds a small section of the DNA double helix and uses one strand as the template to synthesize a complementary mRNA molecule, substituting U for T. It reads the DNA and builds the RNA copy,
Making the messenger.
making the messenger. Then, this mRNA molecule is processed a bit, and it moves out to the cytoplasm. That's where translation occurs, on the ribosomes. It has 3 phases: initiation, elongation, termination.
Okay.
Initiation. A ribosome assembles on the mRNA molecule, finding the start codon, AUG. The first tRNA, carrying the amino acid methionine, binds to this start codon, elongation. The ribosome moves along the mRNA, reading codons one by one. For each codon, the correct tRNA, carrying its specific amino acid, docks in the ribosome. An enzyme within the ribosome catalyzes the formation of a peptide bond, linking the new amino acid to the growing protein chain. The ribosome then shifts over one codon, the empty tRNA leaves, and the next tRNA comes in. It's like a production line.
Adding amino acids one by one.
Exactly, building the polypeptide chain, termination. This continues until the ribosome hits a stop codon on the mRNA. There's no tRNA for stop codons. Instead, release factors bind, the completed protein is freed, and the ribosome components disassemble, ready to start again.
And this can happen multiple times on one mRNA.
Oh yeah. Often, multiple ribosomes translate the same mRNA molecule simultaneously. They form a structure called a polysome, or polyribosome. It looks like beads on a string under the microscope again, but this time the string is mRNA, and the beads are ribosomes.
An efficient assembly line.
Incredibly efficient, making many copies of the needed protein quickly.
It's incredible how organized and precise this molecular machinery is, a true miniature factory. But how do cells decide which genes to transcribe and translate, and when? A root cell doesn't need photosynthesis proteins. A leaf cell doesn't need root hair proteins. How's that controlled?
That's the whole field of gene regulation, and it's absolutely fundamental. It's the reason why different cell types in a plant or any multicellular organism look and function differently, even though they mostly contain the exact same DNA. It's called differential gene expression.
Turning genes on or off?
Basically, yes, turning them on or off or up or down like a dimmer switch. And eukaryotes have evolved really complex ways to do this. One major level of control is chromatin condensation. Remember how DNA is packed?
Yeah, wound around histones, folded up.
Right. How tightly that DNA is packed influences whether the genes in that region can be transcribed. Loosely packed chromatin, called euchromat, is generally active. The transcription machinery can get in there. Tightly packed chromatin, heterochromatin, is largely inactive or silent. The physical wrapping around nucleosomes itself tends to repress gene expression. Things have to loosen up for a gene to be turned on.
So packing controls access.
It's a major factor. And linked to that are histone modifications. Chemical tags can be added to or removed from the histone proteins themselves. Things like acetylation or methylation, these modifications can act like signals, either loosening the chromatin to promote transcription or tightening it to repress transcription.
Changing the packaging locally.
Exactly. Another layer is DNA methylation. Methyl groups can be added directly to some cytosine bases in the DNA sequence itself. This methylation pattern is often associated with long-term gene silencing.
And these chromatin changes, histone modifications, DNA methylation, they don't change the actual DNA sequence, do they?
No, that's the crucial point. They don't change the A's, T's, G's, and C's, but they can be inherited when cells divide, and sometimes even across generations. This is the basis of epigenetic inheritance, heritable changes in gene function without changes in the DNA sequence itself. It's a whole other layer of information on top of the genetic code.
Wow, epigenetics, that's fascinating. What else controls genes?
Then you have specific binding proteins. These are proteins, often called transcription factors, that bind to specific short DNA sequences, usually near the start of a gene. They can act as activators, helping RNA polymerase bind and start transcription, or as repressors, blocking it. It's like molecular switches controlling individual genes or sets of genes.
Turning specific lights on or off.
Good analogy. And recently, we've discovered a huge role for non-coding RNAs. These are RNA molecules that don't code for proteins, things like microRNAs. More NAs. They are tiny RNA molecules that can bind to specific mRNAs and either block their translation or mark them for destruction. It's another way to fine-tune protein production after transcription.
So RNA regulating other RNA.
Yes. A mutation affecting a microRNA in the model plant Arabidopsis, for example, can cause dramatic changes, like severely curled leaves, showing how critical these small RNAs are for normal development.
So it seems eukaryotes have a lot more of this non-coding DNA and many more layers of regulation compared to, say, bacteria.
Oh, absolutely. That's a major difference and a big theme in genomics. Eukaryotic genomes are generally much larger, and a huge fraction of that DNA doesn't actually code for proteins.
What is all that other DNA doing?
A lot of it is regulatory sequences, like those binding sites for transcription factors. A lot is also repeated sequences. You have tandemly repeated DNA, sequences repeated one after another, often found at centromeres, which are important for chromosome movement during cell division, and at telomeres.
The protective caps at chromosome ends.
Exactly, like the plastic tips on shoelaces. In plants, a common telomere repeat is TT2G. You also have interspersed repeated DNA scattered throughout the genome. And a big component of this is transposons, those jumping genes we mentioned earlier.
Still jumping around.
They can. These molecular parasites can make copies of themselves and insert into new locations. They've actually played a huge role in shaping genome size and evolution. Eukaryotes have also evolved sophisticated epigenetic mechanisms, often involving methylation, to silence these transposons and keep them in check, protecting the genome's integrity.
Okay. And wasn't there something about eukaryotic genes being interrupted?
Yes. Another huge surprise was the discovery of introns and exons. Most eukaryotic protein-coding genes are not continuous stretches of code. They are interrupted by non-coding sequences called introns. The coding sequences that actually specify amino acids are the exons.
The gene is in pieces.
Sort of. When the gene is first transcribed into RNA, this initial transcript contains both introns and exons. Then, a process called splicing occurs, right there in the nucleus. The introns are snipped out, and the exons are spliced together to form the mature, continuous mRNA molecule that will be translated.
Wow, extra steps. Why?
It seems complicated, but it allows for something amazing called alternative splicing. Meaning, the exons from a single gene can sometimes be spliced together in different ways. So one gene can actually produce several different related proteins.
One gene, multiple proteins.
Exactly. It vastly increases the coding potential of the genome without needing vastly more genes. It adds another profound layer of complexity to gene expression.
So much processing happens before the mRNA even leaves the nucleus in eukaryotes.
Right. Transcription, splicing, other modifications all happen in the nucleus. Then the mature mRNA goes to the cytoplasm for translation. This separation in space and time, compared to prokaryotes where transcription and translation are coupled, allows for all these extra layers of regulation.
Okay, with all this incredible understanding of how genes work, how DNA is structured and copied, how expression is controlled, well, humans figured out how to intentionally manipulate this stuff. Which brings us to Chapter 10, Recombinant DNA Technology, Plant Biotechnology, and Genomics. This is where biology meets engineering.
Exactly. Recombinant DNA technology, which really took off around 1973, gives us the ability to isolate DNA from virtually any organism, cut it, paste it together with DNA from another source, and insert this combined molecule into living cells.
Combining DNA from different species.
Yes, that's the power of it. It has two huge advantages over traditional breeding methods. First, incredible precision. You can often target and move just the single gene you're interested in. Second, it overcomes natural reproductive barriers. You can transfer genes between species that would never be able to hybridize naturally, say, a bacterial gene into a plant.
Okay, so what are the basic tools for this? How do you cut and paste DNA?
The key tools are really elegant molecular machines. First, restriction enzymes.
Molecular scissors.
Perfect analogy. These enzymes, originally found in bacteria where they defend against viruses, recognize very specific short sequences of DNA nucleotides, maybe 4 to 8 bases long, and they cut the DNA only at those specific sites.
Very precise cutting.
Very precise. And often, they make staggered cuts, leaving short, single-stranded overhangs called sticky ends.
Sticky ends. Why sticky?
Because they're complementary. Any DNA fragment cut with the same restriction enzyme will have complementary sticky ends. So a piece of human DNA cut with, say, E. coli can stick by base pairing to a piece of bacterial DNA cut with E. coli. They naturally want to join up.
Universal connectors.
Exactly. Then you need a carrier molecule, a vector, to carry this foreign DNA into a host cell. Small circular DNA molecules found in bacteria called plasmids are very common vectors.
Okay.
You cut the plasmid with the same restriction enzyme, you cut your gene of interest with it, you mix them together, the sticky ends pair up. Then you use another enzyme, DNA ligase, which acts like molecular glue or tape to seal the gaps in the DNA backbone.
Creating a hybrid plasmid.
Exactly. That modified plasmid containing DNA from two different sources is now recombinant DNA.
Okay, so you've made it. How do you make copies?
DNA cloning. You introduce these recombinant plasmids into host cells, usually bacteria like E. coli because they grow so fast. This process of getting the DNA into the cells is called transformation. Then you grow the bacteria. As they multiply, doubling every 20 minutes or so, they replicate the recombinant plasmid along with their own DNA. You end up with millions or billions of copies of your desired gene. You've cloned it.
But how do you know which bacteria actually took up the plasmid? Transformation isn't usually 100% efficient.
Good point. You need ways to identify the transformed cells. Often, the plasmid vector carries a selectable marker gene. A common one is a gene for antibiotic resistance, like AMPA-R for ampicillin resistance.
So you grow the bacteria on ampicillin.
Exactly. Only the bacteria that took up the plasmid with the AMPA-R gene will survive and grow. The others die. Another approach uses reporter genes. These produce some kind of visible signal. For example, you might insert your gene into the middle of a plasmid gene called LAC-Z. If your gene inserts correctly, it disrupts LAC-Z. On a special growth medium, normal LAC-Z makes blue bacterial colonies, but disrupted LAC-Z makes white colonies. So you just pick white ones.
Visual selection.
Yep. Even more visually, you can fuse your gene of interest to a gene for green fluorescent protein, GFP, from jellyfish.
Making things glow green.
Exactly. You can actually see where the protein you're studying is located inside a living plant cell because it glows green under fluorescent light. Or use luciferase, the enzyme from fireflies, to see where and when a gene's promoter is active. Parts of the plant literally glow in the dark.
That's amazing. Okay, so that's cloning. What if you just need lots of copies of a specific DNA sequence quickly, without bacteria, maybe for forensics or diagnostics?
For that, we have the polymerase chain reaction, or PCR. This technique is incredibly powerful. It can amplify a specific DNA segment millions or billions of times in just a few hours, all in a test tube.
How does PCR work?
It's based on cycles of temperature changes. First, you heat the DNA sample to separate the two strands, denaturation. Then you cool it slightly so that short synthetic DNA pieces called primers can bind anneal to the specific sequences flanking the target region you want to copy. Then you raise the temperature a bit, and a special heat-resistant DNA polymerase enzyme, like Taq polymerase, originally from bacteria living in hot springs, extends the primers, synthesizing new complementary DNA strands extension.
Can you just repeat that?
Repeat that cycle. Denature, anneal, extend 20, 30, 40 times. Each cycle doubles the amount of target DNA, so the copies increase exponentially. 2, 4, 8, 16, 32. You get huge amplification very quickly.
Wow. So versatile. And what about getting a handle on all the genes or all the expressed genes in an organism?
That's where DNA libraries come in. Think of them as collections of cloned DNA fragments. A genomic library aims to contain cloned fragments representing an organism's entire genome, all its DNA, coding and non-coding.
The whole book.
The whole book, chopped into manageable pieces. A complementary DNA, a cDNA library, however, is different. It's created starting from messenger RNA, mRNA molecules, isolated from cells. You use an enzyme called reverse transcriptase, originally from viruses, to make DNA copies of the mRNA.
Working backwards from RNA to DNA.
Exactly. The crucial thing is, since mRNA has already had the intron spliced out.
Ah, the cDNA only has the coding sequences.
Precisely. A cDNA library represents only the genes that were being expressed, transcribed into mRNA in the cells you started with, and it only contains the exons, the protein coding parts. This is really important if you want to express a eukaryotic gene in bacteria, because bacteria don't have the machinery to splice out introns.
Okay, let's bring this technology back specifically to plants now. How is all this recombinant DNA tech and related methods being applied in plant biotechnology?
Right, plant biotechnology is a really broad field, using these tools and others to understand and modify plants. Its roots actually go back quite a way. Think about hydroponics in the 19th century, growing plants in nutrient solutions without soil. That was an early form of manipulating plant growth conditions. This led eventually to modern plant tissue culture. This is the ability to grow plant cells, tissues, or even small organs in sterile laboratory conditions on a nutrient medium.
Growing plants in test tubes.
Essentially, yes. And a key application of this is micropropagation, also called clonal propagation. This allows growers to produce huge numbers of genetically identical copies of a desirable plant from just a small starting piece of tissue or even single cells.
Making clones on a massive scale.
Exactly. It exploits a remarkable property of many plant cells called totipotency.
Totipotency.
It's the ability of a single, mature, differentiated plant cell to de-differentiate and then re-differentiate to develop into an entire new plant, given the right conditions and hormones.
One cell can make a whole plant.
Often, yes. F.C. Stewart famously demonstrated this back in the 1950s with carrot root cells. He showed that cells scraped from a carrot root could regenerate into whole, fertile carrot plants in liquid culture. It was a landmark experiment.
Amazing potential in every cell.
It really is.
Yeah.
Another huge application of tissue culture is producing virus-free plants. Many valuable crops, like potatoes, strawberries, or ornamental flowers, are often propagated vegetatively, using cuttings, tubers, etc., which unfortunately also propagates any viruses the parent plant has.
Passing the sickness on.
Right. But viruses often don't invade or move very slowly into the very tip of the growing shoot, the apical meristem, because the cells there divide so rapidly. So by carefully dissecting out just this tiny meristem tip and growing it in tissue culture, you can often regenerate plants that are completely free of the viruses that plagued the parent stock.
Cleaning up the plant line.
Exactly. This has had a massive positive impact on yields for crops like potatoes and rhubarb.
Okay, so tissue culture is powerful. But genetic engineering offers ways to specifically change the plant's own genes, right? To add new traits.
Yes, that's where recombinant DNA meets plant biology most directly. It allows for very precise genetic modification, often adding traits that would be impossible to get through traditional breeding.
How do you get the new DNA into the plant cells and make it stick?
One of the most elegant methods uses a natural genetic engineer, the soil bacterium Agrobacterium tumefaciens.
That sounds ominous.
Well, in nature, it causes a plant disease called crown gall, basically, tumors. It does this by transferring a specific piece of DNA from its own plasmid, called the tin plasmid for tumor-inducing, into the plant cell's chromosomes.
It injects its DNA into the plant.
It does. This transferred DNA, called tDNA, contains genes that force the plant cell to produce unusual compounds called opines, which the bacterium eats, and also plant hormones that cause the uncontrolled cell growth of the tumor.
A parasite commandeering the host cell.
Exactly. But scientists figured out how to hijack this hijacker. They learned how to remove the tumor-causing genes and the opine synthesis genes from the tDNA and replace them with whatever gene they want to transfer into the plant, say a gene for insect resistance or herbicide tolerance.
Using agrobacterium as a delivery vehicle.
Precisely. You co-cultivate plant cells or tissues with these disarmed agrobacterium containing your gene of interest in the tDNA region. The bacterium transfers the modified tDNA into the plant genome. Then you use tissue culture to regenerate whole transgenic plants from those modified cells.
And the new gene is stable, passed on.
Yes, it becomes stably integrated into the plant's chromosomes and is usually inherited by the offspring just like any other gene, following Mendelian rules. It's a really widely used and effective method.
Are there other ways to get DNA in? Maybe for plants agrobacterium doesn't infect well.
Yes, several other methods exist. Electroporation uses brief, high-voltage electrical pulses to create temporary pores in the plant cell membranes, allowing DNA from the surrounding solution to enter.
Zapping the cells.
Pretty much. Another common one, especially for monocots like corn or rice, which were initially harder to transform with agrobacterium, is particle bombardment or biolistics.
Biolistics. Sounds dramatic.
It kind of is. You coat tiny particles of gold or tungsten with the DNA you want to introduce. Then you use a device, literally called a gene gun, to shoot these DNA-coated microprojectiles at high velocity directly into plant cells or tissues.
Shooting DNA into cells.
Yes. Some particles penetrate the cell walls and nuclei, and the DNA can get integrated into the genome. This method was famously used to create virus-resistant papaya varieties in Hawaii.
Saving the papaya industry.
It literally saved the Hawaiian papaya industry from the devastating papaya ring spot virus in the 1990s. A real success story for the technology.
So what are some other major practical applications we see today?
Oh, there are many now. Pest control is a big one. Genes from the bacterium Bacillus thuringiensis, BT, that produce proteins toxic to certain insect larvae have been put into crops like cotton and corn.
Beet cotton, beet corn.
Right. The plants make their own insecticide, specifically targeting major pests like the cotton bollworm or European corn borer. This has dramatically reduced the need for spraying broad-spectrum chemical insecticides in many areas. Herbicide tolerance is another major application. Genes conferring tolerance to broad-spectrum herbicides like glyphosate, Roundup, have been introduced into crops like soybeans, corn, and canola.
Letting farmers spray weeds without killing the crop.
Exactly. It simplifies weed management significantly, though, of course, widespread use raises issues about herbicide resistance in weeds and impacts on biodiversity, which are important ongoing discussions. We also see work on delayed senescence slowing down aging. For example, modifying cytokinin production in tobacco plants made their leaves stay green and functional longer. This holds potential for increasing yield or shelf life in other crops and improving food quality. Things like developing soybeans with healthier fatty acid profiles, reducing saturated fats, or golden rice engineered to produce beta-carotene, a precursor to vitamin A, to help combat deficiency in regions where rice is a staple.
Okay, so we've gone from single genes to modifying them. But now we can look at the entire genetic instruction set, the whole genome. What is genomics all about?
Genomics is the study of whole genomes, their content, organization, function, and evolution. It's about understanding the entire genetic blueprint of an organism, not just individual genes in isolation.
In a much broader perspective.
Much broader. It encompasses several areas. Structural genomics focuses on determining the actual sequence of the DNA and mapping the genes and other features onto the chromosomes. This involves creating genetic maps based on recombination frequencies between linked genes, like we discussed earlier, and more detailed physical maps based on the direct DNA sequence itself. The ultimate goal is the complete sequence.
Getting the blueprint down.
Right. Then comes functional genomics, which is arguably the bigger challenge. Figuring out what all those sequences do. What is the function of each gene? How do they work together?
How do you even start?
A common first step for a newly sequenced gene is a homology search. You compare its DNA or predicted protein sequence to vast databases of known genes from other organisms. If your new gene is very similar to a known gene, say an enzyme involved in photosynthesis in another plant, it gives you a strong clue about its likely function.
Guilt by association.
Kind of, yes. Another powerful approach is using knockout mutants. You specifically inactivate or knock out a particular gene and then observe what changes occur in the plant's phenotype.
Seeing what breaks when you remove a part.
Exactly. It helps deduce the gene's normal role. And then there are tools like DNA microarray analysis or the more recent RNA-seq. These allow scientists to monitor the expression levels of thousands or even all genes in a cell or tissue simultaneously.
Like a snapshot of gene activity.
Precisely. You can see which genes get turned on or off in response to different conditions like drought, heat, or pathogen attack. Or compare gene expression in different tissues like roots versus leaves. It gives you a systems-level view of gene regulation.
That sounds incredibly powerful. What about comparing whole genomes?
That's comparative genomics. Comparing the complete genome sequences of different organisms provides amazing insights into evolutionary relationships. It's strongly confirmed the division of life into the three domains bacteria, archaea, and eukarya. It's revealed unexpected things like extensive horizontal gene transfer, HGT, in prokaryotes.
Genes moving sideways between unrelated species.
Yes, much more common than previously thought in bacteria and archaea. And comparative genomics has even shown evidence of HGT between bacteria and eukaryotes, like that agrobacterium tDNA transfer we talked about, a natural example of cross-domain gene flow.
So how do plant genomes stack up when compared to others? Any defining features?
Well, as we mentioned, eukaryotic genomes, including plants, are generally larger and more complex than prokaryotic ones. Plant genomes, in particular, often show evidence of ancient whole genome duplications, polyploidy events in their evolutionary past.
So lots of duplicated genes.
Often, yes. And transposons, those jumping genes, have been major contributors to the often massive size of plant genomes. Some plant genomes are truly enormous. What's also interesting is that the number of genes doesn't always correlate neatly with perceived organismal complexity. The small weed Arabidopsis thaliana, which is a key model organism.
The lab rat of the plant world.
Pretty much, it actually has more genes than the fruit fly Drosophila.
Really?
Yep. Model plants like Arabidopsis with its small genome, very fast life cycle, about 6 weeks seed to seed, and ease of genetic manipulation have been invaluable. Rice, or Rhiza sativa, is another crucial model, especially for cereals.
Why rice?
Because its genome was sequenced early, and it turns out there's a high degree of synteny, similar gene order along chromosomes, among all the grasses. So findings in the relatively small rice genome can often be applied to understanding the much larger and more complex genomes of crops like wheat, corn, and barley.
Using the model to understand the crops.
Exactly. Knowing these complete genome sequences is revolutionizing plant breeding. It helps researchers identify genes responsible for important traits like yield, drought tolerance, or disease resistance. These genes can then potentially be manipulated either through advanced breeding techniques or genetic engineering for agricultural improvement.
Okay, let's pull all these threads together now. Heredity, genes, variation, genomes, and connect them to the grand narrative of life.
Yeah.
Evolution. And you can't talk evolution without talking Charles Darwin.
Indeed. His voyage on the HMS Beagle in the 1830s, observing the incredible diversity of life, particularly on the Galapagos Islands, was foundational.
Leading to his revolutionary idea.
Yes, his theory of evolution by natural selection, published in 1859 in On the Origin of Species. It was, and is, perhaps the most influential idea in all of biology. The core idea is elegant. Organisms within a population vary. Some of these variations are heritable. Organisms produce more offspring than can possibly survive. Therefore, individuals with heritable variations that give them even a slight advantage in surviving and reproducing in their specific environment will tend to leave more offspring than others.
Survival of the fittest, but maybe reproduction of the fittest is more accurate.
Exactly. Fitness in the evolutionary sense isn't about being strongest or fastest in absolute terms. It's measured solely by reproductive success, how many viable, surviving offspring you leave in the next generation. Over time, those favorable traits become more common in the population. This differential reproductive success is natural selection, and it leads to populations becoming better adapted to their environment.
What influenced Darwin in developing this?
Several things. He was influenced by the economist Thomas Malthus, who wrote about human populations potentially outstripping their food supply, leading to a struggle for existence. Darwin realized this struggle applied to all life.
Competition for resources.
Yes. He was also deeply influenced by the geologist Charles Lyell, whose work suggested the Earth was incredibly old, far older than previously thought. This gave Darwin the vast timescale needed for slow, gradual evolutionary changes to accumulate.
Enough time for evolution to happen.
Precisely. Darwin also drew an analogy with artificial selection, the process used by farmers and breeders for centuries.
Selecting the best cows or the fastest horses?
Right. Humans deliberately select individuals with desirable traits to breed. Darwin realized nature did something similar, but the selector was the total environment, and selection acted on the whole organism's ability to survive and reproduce, not just one trait. Hashtag tech take two. The gene pool and Hardy-Weinberg equilibrium.
Okay, so Darwin provided the mechanism. How do we think about this process now in terms of the genetics we've been discussing?
We think about it at the population level. We talk about the gene pool of a population.
What's that exactly?
It's the sum total of all the alleles for all the genes present in that population at any given time. Think of it as the population's reservoir of genetic variation. Evolution, at its most fundamental genetic level, is simply a change in the allele frequencies within that gene pool over generations.
So if the frequency of allele A increases and allele A decreases, that's evolution happening.
That's microevolution happening, yes. To understand why these frequencies change, it's actually helpful to first understand the conditions under which they wouldn't change.
A baseline of no change.
Exactly. This theoretical baseline is described by the Hardy-Weinberg law, or equilibrium. It states that in a large, randomly mating population where there's no mutation, no gene flow, and no natural selection occurring, the allele frequencies and the genotype frequencies, like the proportion of AA, and AA in individuals, will remain constant from generation to generation.
The gene pool stays stable.
Theoretically, yes. It's a mathematical principle. It provides a null hypothesis. If a population's allele frequencies are changing, then one or more of those 5 conditions must be being violated. Evolution is occurring. Hashtag tag 3, the agents of evolutionary change.
But those 5 conditions, no mutation, no gene flow, large size, random mating, no selection, they sound pretty unrealistic for natural populations, right?
Highly unrealistic.
Yeah.
They're almost never all met in nature, and that's the point. Evolution happens because these conditions are violated. The factors that disrupt Hardy-Weinberg equilibrium are the agents of evolutionary change.
Okay, so what are these agents?
Let's run through them. First, mutations. As we said, they're the ultimate source of new alleles, the raw material. While mutation rates for any single gene are usually low across a whole genome and a whole population, they constantly introduce new variation.
The source code changes.
Right. Second, gene flow. This is the movement of alleles between populations. Think of pollen blowing from one plant population to another, or seeds being carried. Gene flow tends to make different populations more genetically similar to each other. It can introduce new alleles, increasing variation within a population, but it reduces differences between populations. It can sometimes counteract local adaptation driven by natural selection.
Mixing things up between places. How far does pollen need to travel?
It varies hugely, but studies show that even relatively short distances, maybe 50 to 300 meters, can sometimes be enough to effectively isolate plant populations from significant gene flow via pollen.
Okay, what's next?
Third, genetic drift. This refers to changes in allele frequencies due to random chance events, purely sampling error from one generation to the next. Drift is most powerful in small populations. Imagine you have a rare allele present in just one plant in a tiny population. Just by chance, that plant might fail to reproduce or get eaten, and the allele is lost not because it was bad, just due to bad luck.
Random fluctuations.
Exactly. 2 specific scenarios of drift are important. The founder effect. This happens when a new population is started by just a few individuals, the founders. Their gene pool might, just by chance, have very different allele frequencies than the large source population they came from. Think of a single seed colonizing a new island.
Starting with a non-representative sample.
Precisely. The other is the bottleneck effect. This occurs when a population undergoes a drastic reduction in size due to some random event, a fire, a flood, disease. The few survivors may have a gene pool that, again by chance, is very different from the original population. Certain alleles might be lost, others might become unexpectedly common.
Squeezing through a random filter.
Good way to think of it. Fourth agent, non-random mating. The Hardy-Weinberg model assumes individuals mate randomly, but often they don't. Plants, for example, are more likely to be pollinated by nearby neighbors than by distant individuals. This promotes inbreeding, mating between related individuals. Inbreeding doesn't actually change allele frequencies in the gene pool itself, but it does change genotype frequencies. It increases the proportion of homozygotes, AA and AA, and decreases heterozygotes, AA, over generations. Self-pollination, common in plants like peas, is the most extreme form of inbreeding.
Okay. And the last agent, the big one.
The big one, natural selection. This is the only agent that consistently leads to adaptation populations becoming better suited to their environment. Selection acts on the variation generated by mutation, drift, and gene flow. Individuals with traits that enhance survival and reproduction in their current environment leave more offspring, so the alleles underlying those traits increase in frequency over time.
Driving adaptive change. Can this happen quickly?
It can happen surprisingly quickly, especially when selection pressures are strong. We see it in response to human activities all the time, like plants in heavily grazed pastures evolving to be genetically shorter because the tall ones keep getting eaten before they can reproduce. Or that bent grass, a gross tenuis, rapidly evolving tolerance to heavy metals and mine waste soil in Wales, sometimes within just a few generations. It shows selection can be a potent force, even on human timescales.
Okay, so natural selection is the process that leads to adaptation. Let's unpack that term adaptation a bit more. What exactly does it encompass?
Right. Adaptation can actually mean a few slightly different things in biology. It can refer to a state of being. You might say a cactus is adapted to desert conditions.
Hmm.
It can also refer to a specific trait that helps an organism survive and reproduce in its environment. The spines of a cactus are an adaptation for defense and reducing water loss. The carnivorous leaves of a sundew are an adaptation for getting nitrogen in nutrient-poor bogs.
A feature that helps.
Exactly. And third, adaptation can refer to the evolutionary process itself, the gradual modification of a population over many generations by natural selection, leading to organisms that are better suited to their environment.
The process of getting better suited.
Precisely. And it's crucial to remember, as we discussed earlier, that the phenotype the trait selection acts on isn't just determined by genes. It's the result of interaction between genotype and environment.
Right, the plasticity.
Exactly. Plants often show remarkable developmental plasticity. Their form can change significantly based on environmental cues. Remember the water buttercup leaves? Same genes, different forms in water versus air. Or shade leaves being thin and broad, while sun leaves on the same tree are thicker and smaller. That's plasticity, not genetic difference between the leaves.
Okay. What about patterns of adaptation across landscapes?
When populations show gradual, continuous changes in traits along an environmental gradient, say, plants getting progressively shorter as you go up a mountain, this is called a
A
climb.
gradual trend.
Yes. But sometimes you find distinct populations within a species that are specifically adapted to different local conditions. These locally adapted populations, often with measurable physiological differences, are called ecotypes. Classic studies on Potentilla glandulosa, sticky cinquefoil, in California showed distinct ecotypes adapted to different altitudes, coastal, mid-elevation, alpine. When grown together in a common garden, they retained differences that reflected their origin. Similar things are seen with goldenrod or mountain sorrel strains adapted to different light levels or temperatures.
Locally tuned varieties within a species.
Exactly, demonstrating adaptation to specific local environments.
And what about adaptations involving interactions between different species, like the flowers and the bees?
Ah, yes, that's coevolution. This is when two or more species interact so closely that they act as major selective forces on each other. As one species evolves, it forces the other species to evolve in response, which in turn affects the first species, and so on. It's a reciprocal evolutionary dance.
Driving each other's evolution.
Precisely. The relationship between flowering plants and their animal pollinators is the classic example. Flower shape, color, scent, nectar rewards evolve in response to the senses and behaviors of specific pollinators. And the pollinators evolve specialized mouth parts, senses, or behaviors to efficiently find and exploit those flowers. It leads to these incredibly intricate, often highly specific partnerships.
So all these evolutionary processes, mutation, drift, gene flow, selection, adaptation, coevolution, how do they lead to the formation of entirely new, distinct species? How does speciation happen?
Speciation is the evolutionary process by which new biological species arise. It's fundamentally about populations becoming reproductively isolated from each other.
Meaning they can no longer interbreed successfully.
That's the core idea behind the most common definition, the biological species concept, BSC. Proposed by Ernst Mayr, it defines a species as a group of actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. The key is reproductive isolation barriers that prevent gene flow between different species.
Okay, but you mentioned earlier that this definition can be tricky, especially for plants.
It can be. The BSC works well for many animals, but plants often pose challenges. Many distinct plant species can still hybridize, sometimes even producing fertile offspring if brought together. Remember the sycamores, Platanus orientalis from Eurasia and P. occidentalis from North America, isolated for maybe 50 million years, look quite different.
But they make the London plane tree hybrid.
Exactly, and it's fertile. So under a strict BSC, are there really separate species? It gets fuzzy. Also, the BSC obviously doesn't apply to organisms that reproduce purely asexually or to fossils.
So how else do biologists define species?
In practice, especially for plants and fossils, the morphological species concept is often used defining species based on distinct structural features. The assumption is that morphological differences reflect underlying genetic isolation. There are also various phylogenetic species concepts, which define species based on reconstructing their evolutionary history, looking for the smallest distinct lineages or groups sharing unique derived traits.
Different ways of drawing the lines. Okay, regardless of definition, how does reproductive isolation actually arise? How do new species form?
The most common mode is thought to be allopatric speciation. Allo meaning different, patric meaning homeland.
Separation by geography.
Exactly. A population gets physically divided into two or more isolated subpopulations by some geographic barrier, a mountain range forming, a river changing course, colonization of a distant island. Once separated, they can no longer interbreed. They then evolve independently. Different mutations arise, genetic drift takes different paths, and natural selection favors different traits in their potentially different environments. Over time, they accumulate enough genetic differences that even if the geographic barrier disappears and they come back into contact, they can no longer interbreed successfully. They've become distinct species.
Like the Galapagos finches Darwin saw.
A classic example of adaptive radiation following colonization of islands, likely driven by allopatric speciation on different islands or adapting to different food sources on the same island. The Hawaiian lobeliads are another spectacular plant example. Hundreds of species evolved from likely a single ancestor, radiating into diverse forms adapted to different habitats across the islands.
Okay, so geographic isolation is key. Can speciation happen without that physical separation?
Yes, that's sympatric speciation, SIME, meaning same homeland, new species forming within the geographic range of the parent population. This was once thought to be rare, but we now know it happens, especially in plants, often through polyploidy.
Polyploidy having more than two sets of chromosomes, right?
Right. We discussed diploid 2N and haploid N. Polyploids might be tetraploid 4N, hexaploid 6N, octoploid N, and so on. This can happen in a couple of ways. Autopolyploidy, auto meaning self, involves chromosome doubling within a single species. Maybe an error in meiosis produces diploid gametes instead of haploid ones. If two diploid gametes fuse, you get a tetraploid 4N offspring. This tetraploid might be instantly reproductively isolated from its diploid parents because hybrids between them, triploids 3N, would likely be sterile due to problems pairing chromosomes in meiosis. Hugo de Vries's tetraploid evening primrose, Anatheragigas, was an early example it couldn't successfully cross back to its diploid parent. A new species, basically overnight.
Instant speciation by chromosome doubling.
Potentially, yes, but perhaps more common, especially in plants, is allopolyploidy, allo meaning different. This involves hybridization between two different species, followed by chromosome doubling.
Okay, how's that work?
Imagine two different diploid species, say species A, 2NAA, and species B, 2NBB, hybridize. They produce haploid gametes, A and B. The hybrid offspring is diploid, AB, getting one set of chromosomes from each parent. This hybrid is often sterile because the A and B chromosomes are too different to pair up properly during meiosis.
Can't make viable gametes.
Right. But if a spontaneous chromosome doubling event occurs in this sterile hybrid, either during mitosis in the plant body or through the formation of unreduced diploid gametes, you get a fertile allotriploid, AABB. Now, every chromosome has a perfect homologous partner. Each A chromosome can pair with another A, each B with another B. Meiosis works, fertile gametes AB are produced.
And this AABB plant is a new species.
Yes, it's typically reproductively isolated from both original parent species, AA and BB, because back crosses would produce sterile triploids again. It's a new species formed by combining the genomes of two others.
Wow. Is this common?
Extremely important in plant evolution. Many major crops are allopolyploids. Bread wheat, for example, is hexaploid, 6N, resulting from hybridization and chromosome doubling involving three different ancestral species. Cotton, tobacco, oats, potatoes, canola, many are polyploids. We've even seen it happen in recent history. The Tregopogon, goat's beard species, introduced to North America from Europe, provide a fantastic example. Three diploid species were introduced. In Washington state, scientists documented the formation of two new allotroploid species derived from hybrids between the introduced diploids, all within the last century. It's evolution in action.
Speciation happening right under our noses. Any other ways?
There's also evidence for recombination speciation. This is where hybridization between two species creates novel combinations of genes in the hybrid offspring, and some of these new combinations lead to reproductive isolation from both parent species without a change in chromosome number. The anomalous sunflower, Helianthus anomalous, seems to have formed this way from two other sunflower species.
So shuffling existing genes in a new way creates isolation.
It seems so, yes. And finally, even if hybrids between species are completely sterile, they can sometimes persist and become widespread if they can reproduce asexually, vegetatively. The horsetail equisetum esphericii is a sterile hybrid, but it spreads readily via its underground rhizomes.
Okay, fascinating ways new species can arise. Now, this whole process, evolution and speciation, is it always slow and gradual, like Darwin originally envisioned?
That's been a big debate. Darwin's view emphasized gradualism evolution proceeding by the slow accumulation of many small changes over vast geologic time. However, the fossil record doesn't always show smooth transitions. Often, species appear rather suddenly in the fossil record, persist for long periods showing little change, stasis, and then disappear just as suddenly.
Gaps in the record, or something else.
It could be gaps, but Niles Eldridge and Stephen Jay Gould proposed an alternative model in the 1970s called punctuated equilibrium. They argued that this pattern, long periods of stasis punctuated by relatively brief bursts of rapid change and speciation, might be real. They suggested that most significant evolutionary change might happen relatively quickly, perhaps during speciation events, especially allopatric speciation in small, isolated peripheral populations. Once a new species is established, it might remain quite stable for millions of years.
So long periods of quiet, short bursts of activity.
That's the idea. The relative importance of gradualism versus punctuated equilibrium is still discussed, and the reality is likely a mix of both, depending on the group and the circumstances. But this ongoing discussion just highlights that evolutionary biology is a vibrant, dynamic field, constantly refining our understanding of life's history, something Darwin himself, with his insatiable curiosity, would surely have appreciated. #tagoutro.
Wow, what an incredible journey we've taken today. Seriously, from the fundamental units of heredity, those genes on chromosomes,
The double helix.
all the way to the grand scale of species formation and the engine of evolution. We've seen how DNA's elegant structure allows it to be copied, how its coded messages get translated into the proteins that build the plant.
And how that whole intricate system is so finely tuned by all those regulatory mechanisms, chromatin packing, epigenetics, non-coding RNAs. It's incredibly sophisticated.
It really is. And we explored how understanding these processes empowers us, how we can use tools like recombinant DNA technology and plant biotechnology to, well, to address some really significant real-world challenges.
Absolutely. From improving food security and nutritional value to developing pest and disease resistance, reducing pesticide use. From Mendel's peas figuring out the basics to literally making plants glow to study gene expression, the story of plant genetics is just this amazing testament to nature's ingenuity and, our growing ability to interact with it at the deepest molecular level.
So, thinking about all this, what does it mean for you, the listener, as you continue to explore the world around you? Here's where it gets, I think, really interesting, maybe even a bit profound. Given our increasing ability to precisely modify plant genomes using tools like CRISPR now, even more precise than what we discussed,
Right, gene editing.
and considering the accelerating pace of environmental change, climate change, the need to feed a growing global population, how might our role, humanity's role as natural selectors, evolve?
That's a powerful question.
Are we in essence becoming co-creators of plant evolution, moving beyond just artificial selection to direct genetic modification on a massive scale? And if so, what new responsibilities does that entail for the future of the planet's biodiversity, for agriculture, for food security?
A very powerful thought to ponder next time you're looking at a field of crops or even just a houseplant, thinking about its genetic past and perhaps its engineered future. We really hope this deep dive has given you a fresh perspective on the complex, dynamic, and truly beautiful world of plant life. Thanks so much for tuning in.
From all of us here at The Deep Dive, we really appreciate you joining us for this exploration. Until next time.